Okay, this is the last lecture in the series about brain imaging using FSL, and today we're going to talk about the statistical thresholding options that you've got in feet. These images I've got on the first slide here represent the same contrast. It's actually one of the contrasts you did in the week 2 workshop. So we've got here the visual contrast versus baseline, and each of these three is the same data but thresholded at 3 different thresholds. So the top image has the most liberal threshold, the middle image intermediate, and the third image a kind of strict version of thresholding in which family-wise error is strictly controlled. And you can see that in this third image there are far less red and orange areas, which are the voxels that are declared active, than there are in either of the above 2 levels of thresholding. These bottom 3 images give you the same thing for the auditory threshold that you ran in the week 2 workshop. With this relatively liberal threshold, you can see you've got a big blob for the auditory cortex around here. You've also got quite a bit of noisy stuff scattered around. As you increase the strictness of the threshold, you can see that the area activated shrinks until it's really only on this one slice for the strictest threshold. Obviously today we'll be looking at the different thresholding options and getting some idea of how to make the difficult choice of what's an appropriate threshold level. This is just an overview slide. We'll remind ourselves what it is that we're performing the thresholding on, i.e. the Z-stats. We'll remind ourselves of the details of the multiple comparisons problem, which is the main issue you're having to deal with when you're choosing your thresholding methods in fMRI. We'll cover several different approaches to dealing with that multiple comparisons problem, and we'll finish by taking a slightly different perspective, looking at the issue of reproducibility of results from fMRI experiments. So rather than focusing on controlling the type 1 error rate, which is what you're doing when you're thresholding, we'll focus on what you need to do to make the experiments more reproducible. In today's workshop, we've got a worksheet on how to write batch files in Linux to call the FSL programs, which can save you an awful lot of time and allow you to do things that you probably wouldn't otherwise think of doing if you were just using the graphical user interface. I'll give you some more information about the assessment. And also you can finish the worksheets from last week and the week before if you didn't finish those. So what is it that's being thresholded? And of course it's the value of a statistic, a Z-stat generally. Before we can threshold our images, we have to quantify the probability that the activation level at a particular voxel is just due to the unreproducible random variation in the data, and that it just happens that that random variation looks like the time course of one of our explanatory variables. So what's the probability that that's happened? To get to that probability, we first calculate a statistic which has a known probability distribution for the special case where the only variation present in the data is the unsystematic random variation, i.e. this is the null hypothesis. T-stat and Z-stat, they both have known distributions when the null hypothesis is true, and therefore can be used to calculate the probability of a particular data set being obtained if the null hypothesis was true. So your null hypothesis for your simplest contrast of parameter estimates, which is just equal to the parameter estimate for a single explanatory variable from the design matrix, that null hypothesis is that the true mean value of the parameter estimate in the population is 0, with of course some standard deviation, some variability around that. Now, for small samples we expect them to have means that are different from zero due to the random sampling process and the fact that they're small samples. On this slide we'll take a look at how the actual Z statistic is formed, which we then apply the thresholds to. Let's consider the case of the simplest first level contrastive parameter estimates, which is just equivalent to parameter estimate minus 0, so it's the same as the parameter estimate for a particular explanatory variable from the design matrix. And here we're assuming a reasonably large sample size, where sample size is time points, but this assumption is always met. First level time series data in fMRI, basically the fact that t equals z once the degrees of freedom are large enough. Now, so our numerator is the parameter estimate. Obviously, a larger parameter estimate clearly implies that a particular explanatory variable is making a bigger contribution to explaining the variation in a voxel time series. So we divide that parameter estimate by the value of the value for that voxel at the varcope image. So the varcope image, as you should know by now, contains the values of the residual variation at each voxel. That is the residual variation that couldn't be explained by any of the explanatory variables in the design matrix. Now, dividing the varcope by the square root of the TDOF value is something we do in order to take into account the fact that, all other things being equal, longer time series produce more reliable results. So remember the TDOF, that contains a voxel-wise estimate of the number of independent observations in a time series, which is somewhat less than the number of actual data points in the time series due to the autocorrelation problem. So the nature of this formula is that in order to halve the value of the denominator here, which would double the value of z, you would need to quadruple the sample size, or TDOF, the sample size being time points. So this calculation is performed separately at every voxel when you produce a z-stat image, and of course independently at each voxel from all the other voxels, because this is a mass univariate approach. On this slide we'll consider two other cases in which we can calculate a z statistic at each voxel. One of these is if your contrast of parameter estimates is of the 1 minus 1 type rather than the 1 minus 0 type, then you would expand the denominator, the standard error part of the formula, to use the varcope data from both individual parameter estimates that we use to make the cope, and the numerator on top would just be the value of parameter estimate 1 minus the value of parameter estimate 2 at the voxel of interest. Now, if you're producing a z-stat image at the second level or higher level for group analysis, then the TDOF is no longer equal to the number of time points in the data. The TDOF is going to be equal to the number of participants in the data. The varcope now represents between participant variation that couldn't be modelled by anything in the second level design matrix, and of course this is all basically true for the ordinary least squares option for second level analysis in feet. If you're using the flame mixed effects option, then actually it's more complicated, and calculation of the z-stat uses both the var codes from the first level and the second level, and also the teed off images from the first level and the second level. Now let's just take a look at Z-stats again. What we're doing in the previous few slides is calculating Z-stats. So this graphic shows us the most important properties of Z-stats from our point of view, which is that a Z value, if we consider a Z value of 1 first of all, then here's Z of 0 in the middle, and for a Z value of 1 we know in advance that 34.13% of Z-stats will fall between 0 and 1, and likewise it's symmetrical, so 34.13 of Z-stats will fall between 0 and -1. So this is what we'd expect under the null hypothesis. We would expect this much variation. The total proportion of Z-stats that fall between -2 and 2 is about 95%. So if an actual Z-stat is larger than 2 or less than -2, then it's only 5% likely we could get a value that large by random variation, random sampling effects. So at that point we, in our behavioural research at least, tend to reject the null hypothesis and say we think that it's false. Okay, so that's the distribution of the statistic we're calculating in the case where there is no effect of the experiment, essentially. On this slide we'll consider a kind of simple thresholding you can do in a subjective way using FSL view. So in this image at the bottom, that's just an image of the value of Z across a particular axial slice in the brain, where the yellow values are higher values of Z and the darker red ones are lower values of Z. So there's a Z statistic at every voxel there, producing this heat map when we don't apply any threshold to that. Now the process of applying a threshold is basically one of taking something that varies in a continuous way, such as the Z values in this image in the slice on the left. They vary continuously across the brain, producing that heat map. Thresholding is just converting that continuum into a binary cutoff where values below a certain point are rejected, thrown away, and values above that point are considered to have passed the threshold. So obviously it's going to be problematic. There's never going to be a perfect threshold value to apply to a continuum. In essence, you're always going to lose some information about the data by doing thresholding. But there are reasons why we need to do it, of course. So let's consider what happens if we put a threshold on this image in FSL view such that it only shows Z values greater than 1 and overlays them on the template image underneath. You can now see that you start to see structure in the activation, and some parts where the Z numbers were lower have just been removed. Now statistically, a Z value that's larger than 1 occurs 16% of the time when there's no signal in the data. That's not considering values of Z that are less than 1 as well, it's just the ones greater than 1. In other words, on that previous slide, 16% of the area underneath the curve corresponds to values greater than 1. If we do the same thing but with a Z threshold of 2, what we're doing now is we're saying we're only prepared to show values on our heat map where that would occur 2.3% of the time were there no signal in the data, because only 2.3% of the area under the curve corresponds to z-scores greater than 2. And down here in the image we see that these blobs are now getting somewhat smaller, but they're not a lot smaller by moving from Z1 to Z2. If we show only Z values greater than 3, then in a set of data with no actual signal other than random variation, that correspond only 0.13% of the area under the curve. And now the activation starts to look like this, on the one on the right, where you can see that it's quite restricted. I just wanted to mention on the previous slide that, of course, negative values of Z are just ones where the value of cope is negative rather than positive. Another important thing to bear in mind about what was discussed on the previous slide is that Z threshold corresponding to the proportions of area under the curve, which are the p-values which I discussed, those are just being performed voxel-wise. Each voxel is being considered completely independently from the others, as if it was the only voxel in the brain. Now, the actual choice of threshold is referred to as the alpha level, and is the probability you are prepared to accept that your result is noise rather than signal. Now, conventionally, in the behavioural experiments that you're more familiar with, an alpha level of 0.05 is often adopted, which corresponds to Z values that are greater than 1.96 or less than greater than 1.96 or less than minus 1.96 for a two-tailed hypothesis. In other words, 5% of the area under the Z curve is accounted for by values more than 1.96 or less than minus 1.96. Now, the reason by convention that 0.05 has become a common choice of alpha level is that it gives you a good compromise between your type 1 error probability, the probability that your positive result is actually the result of random sampling effects, versus the other type of error, the type 2 error, which is that there is a real effect in the population, but that you fail to report it. In other words, the real parameter estimate in the underlying population is larger than 0, but perhaps the random sampling effects in this particular experiment make it appear smaller than it is in reality, and you miss it. And obviously, as you use stricter and stricter alpha levels, for example 0.01 instead of 0.05, it becomes easier to miss real results. You get more type 2 errors. So that's why we don't use a stricter alpha level than 0.05 normally. But we've got a big problem in neuroimaging in that we're actually performing our calculation of Z and our thresholding at every voxel, and so given that if we use the 0.05 alpha level, it would be a 5% chance at every voxel of a false positive result being reported. So we'd actually get lots of false positives across the brain. We need to do something about that. So to look a bit more formally at the multiple comparisons problem, if we had an image where we only had noise, no experimental effects at all, and we thresholded it using a voxel-wise threshold of Z having to be greater than 1.96, i.e. if we also allow Z values of less than minus 1.96, then that corresponds to thresholding at a p-value of less than 0.05. Then if we have 200,000 voxels, we will get 0.05 multiplied by 200,000 equals 10,000 false positive voxels surviving thresholding. those false positives might therefore be mistaken for activation of interest in a contrast. Now, while that's obvious theoretically, there's quite a famous paper that kind of did a practical version of this, where they actually scanned dead salmon in a social psychology experiment with an experimental manipulation that would be of interest if the participant was human and alive, and then they performed all the statistics and thresholded the functional contrast of interest at the 1.96 Z threshold, and they got activation in the salmon's brain and brainstem for some social psychological contrast. And this is a published paper and made a lot of people think about thresholding in fMRI. Actually won the Ig Nobel Prize for that. Now, one solution that you might be familiar with from your behavioural data analysis with SPSS is to control the family-wise error rate, where family equals the number of tests being performed. And given that what you want to do is to control alpha, and you want to maintain alpha perhaps at 0.05 across all the family of tests that you're performing, then that's one way of controlling your false positive rate. So if we look at doing that, which you may be familiar with the name for that, that's the Bonferroni method. We divide our risk of reporting a false positive among all the tests we're performing. So with 200,000 voxels, we'd go alpha divided by 200,000 equals 0.00000025, and that p-value corresponds to a z value of z greater than 5.026 for the one-tailed case where we're only interested in positive differences. Now, z-scores greater than 5 are very unusual, so this is going to be a very conservative test. And you'll probably have a very high, well, you will have a very high type 2 error rate if you apply that kind of Bonferroni correction. In many data sets you would miss all the activation in the data. So this, of course, is making the assumption that the family of tests is equal to the number of voxels in the brain, i.e. that all the voxels are independent from each other. And that assumption is plainly wrong for fMRI data because there is spatial structure in the brain. Bits of the brain that process similar types of information tend to be located adjacent to each other. So we have to find a more appropriate way of controlling family-wise error rate in fMRI, which takes into account the non-independence of adjacent voxels. This is a bit like the problem we had with the non-independence of adjacent time points in the time course, which we corrected for by estimating the autocorrelation structure and turning it into a filter. We need also spatially to estimate the true number of independent comparisons we're making in a set of fMRI data. This is where random field theory briefly enters our story, which is a way of assessing the extent to which there is spatial structure in the data, and therefore the extent to which it differs from just the total number of voxels in the data, i.e. a way of assessing the real number of functional voxels, if you like, rather than the actual number of voxel time courses. So the image in the middle is just a slice from an EPI functional image, not smoothed, but you can see that although it's not smoothed, there is clearly structure in this data, isn't there? The ventricles are the most obvious bit of structure there, but you've also got other recognizable non-random variations in the spatial image intensity. On the right is the same image after some spatial smoothing has been applied, and you can see that increases the smoothness of the image and reduces the kind of granularity. On the left is an image that represents the number of statistically independent units in this slice of data, estimated using random field theory. So what would happen is, given this estimate of the statistically independent number of units, and those are referred to as results, resolution elements, you'd end up doing something like a Bonferroni correction, where you divide alpha by the number of observations, where the number of observations is the number of squares in this image on the left, rather than the original number of voxels from the image in the middle. So, as I said, a result is a resolution element. Now, a resolution element is defined as the full width half maximum of the smoothness of the image. So this full width half maximum concept we encountered before, it's the y-axis value of a Gaussian at half its full width. So now the smoothness of the image is characterized using Gaussians, and I'm not going to get into the details of this theory because it's quite complicated, but the result size is that full width half maximum of the smoothness of the image, and it's sort of similar to, but not exactly equal to, the number of independent observations in the image. It's an estimate of that, effectively. So therefore we're basically correcting our statistics for the number of results rather than the number of voxels. And a really important thing to note if you're using this method, which is the voxel option for correction of multiple comparisons in feet, if you're using this method, it really makes a lot of assumptions about the smoothness of the image in order to fit these Gaussians to it, and so it's going to work okay at the first level, but at the second level it only works well if you've got quite a large sample size or if you've smoothed your data a lot. In the next few slides I'll take you through the different options that are available on the Post-Stats tab in feet for control of false positive rate. So the FWE, or family-wise error option, that's actually selected by selecting the option called voxel on the Post-Stats tab, and that's what I was talking about on the last couple of slides really. If you select this option, you are setting the alpha level that controls the probability of one or more false activations occurring in the whole brain image. So the value you set there is the probability that you accept of there being one or more false activations in the image. What you're doing is you're controlling for a number of tests which is effectively equal to the estimated number of results in the image. Works better when the images are smoother. If the images are not very smooth, the assumptions may not be met, and so particularly for group analysis with relatively small sample sizes, there may be insufficient smoothness in the data for the FWE option to be a good idea. If you choose the uncorrected option on the Post-Stats tab, then what that means is uncorrected for making multiple comparisons. You are still able to set an alpha level for each voxel, so therefore at each voxel you have a certain probability that your result is false positive. The other option in feet is to select cluster based thresholding, which we'll spend a little bit of time talking about now. So imagine you use the uncorrected option and set an alpha of something like 0.01 and look at the results. You will see various clusters or blobs in the images, and some of those clusters are very small, comprising just 1, 2 or 3 individual voxels, whereas other clusters are more like big blobs consisting of hundreds of adjacent voxels. Now you know that you haven't taken into account the number of voxel-wise comparisons, so with an alpha of 0.01 you'll definitely have some false positive activations in the image, maybe 1 in 100 voxels are falsely activated. But cluster thresholding takes advantage of something which intuitively is easy to understand, which is that those very small activation clusters are much more likely to have arisen due to the noise in the data happening to look like the shape of one of your regressors over time than the large clusters are. It's very unlikely, isn't it, that a lot of voxels will independently all happen to be false positives and happen to be adjacent to each other in the image to form a big blob. Those random activations are much more likely to be isolated. Now there is one exception to that rule, which is that you may be particularly interested in and have a priori predictions about a region that is a functional unit in the brain but which is known to be small. For example, the superior colliculus. So even if you manage to activate the whole thing, it's only a few voxels. Right, if you choose the cluster option, then you will set actually 2 alpha values. And in my experience, most people who use this method of thresholding actually don't know what those 2 alpha values mean. So let's take a moment to look at this. The first alpha value is just an uncorrected for multiple comparisons voxel-wise threshold. It's exactly the same as if you chose the uncorrected option in feet. And that's there just to get an initial set of clusters. And usually you set that fairly liberal, equal e.g. alpha 0.01. And the second alpha you set is the cluster threshold. And that's the one where you're actually taking family-wise control over the false positive rate, but you're doing it at the cluster level. So given the first threshold and the clusters that causes to appear in the image, you then define a probability that there are 1 or more false positive clusters in the image, which is the probability that you're prepared to accept of there being 1 or more false positive clusters in the image. Based on that, a critical value is calculated, which is the size which the size that clusters have to be or bigger before they are considered significant clusters, given your desire to control the probability of a single false positive cluster at a certain level. And of course, with this method, other things being equal, for a larger cluster the achieved significance level will be higher. Let's try to compare now a voxel-wise approach to thresholding with a cluster-wise approach. So this diagram here shows a strip of voxels along the space dimension, a one-dimensional strip. So imagine taking a single coronal slice of the brain, but then within that coronal slice just picking one particular value of y, one particular height on that coronal slice, and just taking a strip of voxels from that coronal slice at that particular y value. Okay, so that's where the data comes from, and the data points are z-stat values. Okay, so as we move across our strip, the z-stat values go up and they go down, and you can notice on here that I've indicated with 2 green arrows the position of 2 potential brain activations. One of them is relatively small but has very high Z values, and the other one is much larger but doesn't achieve such high Z values. And intuitively they perhaps look about equally plausible as activations. Now if we apply voxel-wise thresholding to this strip of Z values using a value of Z critical value of Z to correspond to the alpha level we want, then that would perhaps result in all of the voxels below that black line being declared non-significant, and the voxels above the black line being declared significant. So this method of thresholding voxel-wise is likely to detect the small cluster that's highly activated, but not detect the other cluster, and that's potentially a false negative, a type 2 error. And that would be the sort of thing would happen if you used a typical kind of voxel-wise threshold. When people are doing a voxel-wise threshold, they often use 1 in 1,000 as being a reasonable value to choose. If you're not going to explicitly correct for multiple comparisons, you want to be quite strict about your individual comparisons. Now let's take a look at cluster-wise thresholding. As I said earlier, you've used 2 actual thresholds to achieve this. What's drawn on here at the moment with the black line is a voxel-wise threshold that is used to first define the clusters that are submitted to thresholding, and it's set a bit lower than if you're only using voxel-wise thresholding as your only method of thresholding. So here the voxel-wise threshold would initially permit 2 clusters to go forwards, the small, spiky, high intensity cluster and the larger but lower intensity of activation cluster both go forwards to the cluster stage. And then the next thing that has to happen is to calculate a critical value of cluster size to use to decide whether individual clusters pass thresholding or not. And as I said earlier, it's the probability value for a larger cluster is lower, i.e. larger clusters are less likely to occur by chance. So if we want to, let's say, have a probability of only 1% that a cluster is a false positive cluster, then we might set the threshold like this here. We might set it that voxels have to be voxel clusters have to be this large in the space dimension before they're declared active, and based on that critical value, this large cluster passes the threshold, but this smaller one doesn't. So using this method of thresholding, you might manage to reverse the pattern we saw on the previous slide. You might manage to declare the other blob active, but not the one that was declared active on the previous slide. Now, cluster size, K critical, that corresponds to a particular value of K alpha, the probability you want to accept of a false positive cluster in the image, depends on the smoothness of the image, and resols are used to calculate this. So once again, if you're using this method of thresholding, it's a good idea to have fairly smooth images, and that can be achieved either with quite large sample sizes at the second level or by applying quite a bit of smoothing to your first level data. And it's usual to set a fairly liberal voxel-wise alpha when using cluster thresholding as the initial thresholding step, unless you are trying to use cluster thresholding to look for small activations like ones in the superior colliculus. Then you tend to set it a bit higher. Okay, this slide illustrates how the number of clusters identified in the image will depend on that initial choice of voxel-wise Z threshold. Here the same strip of voxels with varying Z values, the variation in the vertical position of the pink line, is thresholded at 3 different levels, and this top one would result in 1 cluster moving forward to the next thresholding stage. If you choose this threshold here as your initial threshold, 2 clusters go forward. If you choose this lower initial threshold here, then only 1 cluster goes forward again, obviously 1 large cluster. The other thing that determines how many clusters are going to be found initially, other than the choice of voxel-wise Z threshold, is the amount of smoothing that you apply in your pre-processing. Here's a table to illustrate more formally the dependency of the actual cluster size threshold that will be chosen on that voxel-wise threshold. So what I'm saying here is that the two thresholds interact with each other. The actual ultimate value of K critical that corresponds to a particular specified alpha level is going to vary depending on the initial height threshold that you applied to initially determine the set of clusters to send forward to the actual thresholding procedure. And this sort of complexity puts some people off using the cluster-wise thresholding method. So if we choose an initial voxel-wise alpha of 0.01 corresponding to a Z value of 2.33, then because that will send forward more clusters, then the critical value for a cluster to have, the critical value in order to get only a 5% chance of one of those clusters being a false positive is that the clusters have to consist of 310 voxels or larger, so they have to be big clusters. So if you set your initial voxel-wise alpha when you're doing the thresholding procedure, if you set that low, then at the second stage it's going to determine that you need big clusters to pass the critical value, and so you're going to miss small activations. If instead we go in with an initial height threshold of 0.001, i.e. an initial Z value thresholding of anything above 3.1, then this time to get only a 5% chance of the cluster being a 5% chance of the cluster being a false positive, it only has to consist of 93 or more voxels. And then if we go a bit even stricter on the initial height threshold, we put that in at a Z of 3.72, then ultimately voxel clusters of 32 voxels or larger can have this property that they've got less than a 5% chance of being a false positive cluster. So that would be the kind of settings we'd use if we were looking for a region like the superior colliculus using this method of thresholding. I should say these values here depend on a particular data set. I generated these by using a particular data set, but you would get similar values from other data sets. Let's take a quick look at other thresholding options. There are non-parametric approaches which are based upon permutation testing, and permutation testing involves permuting or shuffling the data. Perhaps it's easier to imagine this as a kind of second level t-test design, where you've got one group of participants in one experimental condition and another group in the other. If we shuffle the labels, i.e. we randomise whether participants are allocated to group A or group B, ignoring their actual real allocation to group A or group B, and then we compute means and standard deviations, etc. And these are, we know that we've made the null hypothesis true, in a sense, in our shuffled data. And if we calculate all the possible shuffles that there could be of the data and all of the statistics for means and standard deviations for each set of shuffles, then we can build up a distribution of what the results look like when the null hypothesis is true, and therefore what percentage of them have real differences that are equal to or exceed the one we observed with our actual group memberships. So that approach can now be used in neuroimaging. It is quite computationally intensive and tends to take a while, but it is a good idea under some situations such as small sample sizes. Another option is to use the false discovery rate as the parameter that you control instead of controlling family-wise error rate. So remember, family-wise error rate is the probability of the full set of voxels containing one or more false positives. If we instead control the false discovery rate, we try to control the proportion of the voxels that are declared active which are falsely declared active. So if we choose a false discovery rate of 0.01 and we're declaring 1,000 voxels active, then on average across many repetitions of this experiment, 10 of them would be false positives. Note, there's no guarantee that in this particular experiment you've got an exact false discovery rate of 0.01. The guarantee is that on average across many repetitions of the experiment, that would be your false discovery rate. If you are only declaring 200 voxels active and you've set false discovery rate to 0.01, then that's approximately 2 false positives. So nice property of this way of dealing with the false positive rate is that it scales with the amount of activation that you're actually finding. And that there's a lot of common sense about that, isn't there? We don't mind quite a few individual false positive voxels if we've got very large, strong activations. But if we're only got small, very small focal activations, we really don't want many false positives in there at all. It also works better as a method using unsmoothed data. So again, that recommends it for small brain areas that are going to be hard to find in heavily smoothed data. To use false discovery rate in FSL, you need to use a command line program. This is a technique that is potentially very useful, but do read about it because there are one or two caveats and questions over its validity. Another thing you can do to try and improve your statistics is the use of a brain mask, which basically is a mask that attempts to isolate just the grey matter or just the parts of the brain you're interested in, thereby reducing the number of voxels being considered and hopefully easing the multiple comparisons problem. In general, we're only expecting to see activations in grey matter, not white matter or cerebrospinal fluid, ventricles, etc. So you might as well remove them from the image if you can. You then go on to use a family-wise error rate control or a false discovery rate control as before, but you avoid the problem of them becoming excessively conservative due to the large number of voxels in the image. So if you want to remove white matter ventricles, you first perform a segmentation on your high-resolution anatomical image, and with the functional data co-registered to that structural image, you're able to remove the non-grey matter voxels from the image prior to fitting your design matrix. Now, if you are going to do this, then it's probably a good idea to first run the analysis with all your voxels in and then run it again after the brain mask has removed the non-brain voxels, because sometimes you pick up activation in places where it really shouldn't occur, such as in the white matter or the ventricles, and those false activations can be a clue to their presence of head motion problems or image spikes that you might then want to do something about. Another thing you can do is consider scanning only the part of the brain that you're interested in. Have less slices, position them in such a way obliquely that only covers part of the brain. You'll also at that point be able to have a shorter TR, therefore more time points in your data, or smaller voxels. If you do that, then also do acquire one whole head EPI image, as it helps for registration. Another approach is just to extract a region of interest from the whole brain and do separate analysis of that particular time course, perhaps using the tools in FSL for it, such as feet querying. Right, for the final part of this lecture, I'll talk about a slightly alternative perspective on the thresholding issue. Genovese, Lazar and Nichols were quite respected neuroimaging methodologists, have gone down on record with this quote: "Variation across subjects has a critical impact on threshold selection in practice. It has frequently been observed that, even with the same scanner and experimental paradigm, subjects vary in the degree of activation they exhibit in the sense of contrast to noise. Subjective selection of thresholds set low enough that meaningful structure is observed, but high enough so that appreciable random structure is not evident, suggests that different thresholds are appropriate for different subjects." And that was a strategy that I suggested some of you use in the workshop last week when examining individual participant data. Set the threshold so that you see meaningful structure, but you don't see that kind of salt and pepper randomness, and that will cause a threshold to vary across participants. I've seen papers published by Jodie Cullum using this strategy. So maybe we should be using intuitive thresholding after all. Sometimes thresholding practices can perhaps be negative for the overall progress of science. Journal reviewers and editors are always reassured if the rate of false positives has been controlled using strict family-wise error control of the Bonferroni type to deal with the multiple comparisons problem. And so researchers produce all their efforts to produce activations that survive such stringent tests. But as I said earlier, there's a trade-off between controlling that false positive rate in the data and the more hidden false negative rate, the activations that are missed. And it might be that use of family-wise error rate procedures is producing the wrong balance between the two types of error that an experiment can make. This issue has been examined in most detail by Tyrion in 2007, who looked at reproducibility of brain imaging results. The thing is, when you've only got one set of data, which is the position we're generally in when we're trying to publish something, we can use classical procedures to give us control of the false positive rate. But this doesn't quantify the probability that there is a real effect in the population which is not being reflected in this specific sample due to chance sampling events causing the effect size which to be smaller in your sample than it is in the actual population, and therefore easy to miss, particularly with small samples. So what we want really is to be able to repeat an experiment many times and find almost identical activations each time, because that would imply that both the false positive rate and the false negative rate are low. If we repeat the experiment and each time the activations we get are slightly different, that could be due to the presence of false positives, false negatives, or a mixture of both. So assessing reproducibility would provide a way of knowing something about how many real activations are actually being rejected by thresholding the usually ignored false negative rate. Tyrion scanned 80 people using a number of standard localizer paradigms, for example a motor cortex localizer. The motor cortex is localized by finger tapping. They then could randomly select subsets of their data. For example, they could randomly select a sample of 20 from their overall sample of 80 and recompute the analysis. And they can do that for another sample of 20. You can do it for all possible samples of 20, a bit like a Cronbach's alpha procedure. And they can see how much variation there is in the results when sample sizes are of 20, and they can do that again. They can see how much variation there is between the results of analysis for sample sizes of 10 or 30, as well as varying the sample size and repeating the analysis. They also tried out different statistical thresholding methods. Therefore, their results will indicate the effects of sample sizes as well as thresholding methods on the reliability or reproducibility of fMRI results. Now, somewhat surprisingly, they found that reliability of fMRI, that is, the ability to reproduce findings, was best when the alpha value selected was between 0.0035 and 0.001, uncorrected for multiple comparisons. So just setting a 1 in 1,000 chance that any individual voxel time course will give you a false positive result and produce better science than all of these complicated procedures for trying to fix the overall rate of family-wise error across the data set. So allow 1 or 2 out of every 1,000 voxels in the brain to be declared active incorrectly. That actually gives you the best trade-off between the false positive rate and the false negative rate. And maybe obsessing about controlling the probability of a single false positive in the whole data set is not a good thing in brain imaging. Thirion also found that the non-parametric permutation-based methods were more reliable than their parametric, more commonly used parametric versions. For the second level type of analysis, then carrying variance estimates forward from the first level as well as the effect sizes did improve reliability, so the FSL team are correct to advocate mixed effects analysis as embodied in the flame option rather than using ordinary least squares. The cluster-wise method of family-wise error control was more reliable than the voxel-wise method when group analysis was the objective. They also observed that high random effects stats values, that is high values in the COPEs and in the Z-stats, tended to coincide with the highest areas of between participant variance, which they suggested may be due to poor spatial registration between participants. Now, the biggest question of all perhaps was how big does the sample size need to be for a neuroimaging experiment in order to get a result that another group of researchers should be able to replicate. And that number they estimate is somewhere between 20 and 27. You know, it's a sobering thought that many fMRI studies are using between 10 and 14 participants. Right, that was the end of the lecture. We may now read or discuss something about these thresholding issues such as those raised by Thirion, but in the workshop we'll look at shell scripting if you haven't already got things to catch up on from previous weeks. Shell scripting is something that can save you a lot of time if you're prepared to put in the initial effort to learn how to do it. As well as saving you time, it gets you into using those parts of the FSL software that are only available via the command line. So, for example, FSL Maths is a really useful program, but it doesn't have a graphical user interface. It's much more efficient to call these programs through a script because you can then save the script as a text file. You haven't got to enter the commands again in the future, and the script can do the same thing to many participants or many sessions of data all at once, not having to repeat yourself. So we'll do the scripting exercise that the FSL group at Oxford provide, and I've just put some helpful hints and tips for that on Blackboard, and I've printed that out as well.